Imagine your body's DNA is a massive instruction manual. It is incredibly detailed, spelling out how to build and operate you using a code of 3 billion letters. Usually, this manual is copied perfectly, but sometimes a single letter gets written down wrong. That tiny spelling mistake is the cause of a rare condition called Snijders Blok-Campeau syndrome. This condition traces back to a specific set of instructions called the CHD3 gene. Think of CHD3 as a manager in the brain. Its job is to unpack tightly coiled DNA so the rest of the body can read it. But in people with this syndrome, the code has a typo. Right where there is supposed to be a G, the manual has an A instead. Because of that one incorrect letter, the manager can't do its job. The DNA stays packed away, and crucial instructions for brain development go unread, leading to difficulties with speaking, learning, and basic motor coordination. To find a cure, scientists first had to replicate this exact genetic typo inside the brains of laboratory mice. This typo caused the mice to struggle with coordination and social communication, mirroring what doctors see in human patients. We cannot use standard drugs to fix this, because they only mask the symptoms. To actually solve the problem, we have to go into the living brain and fix the broken letter at the source. This requires an incredible level of biological precision, rewriting the code of life inside a living, thinking animal. Traditional gene editing acts like molecular scissors. It is designed to cut right through the DNA strand to remove or change a section. But using scissors inside a living brain is risky. When you cut DNA, the cell has to repair the break, which can cause unpredictable and messy damage. Instead of scissors, researchers developed an adenine base editor. It works less like a cutter and more like a molecular pencil with an eraser. This tool chemically erases the incorrect A and precisely writes in the correct G without breaking the DNA strands. This allows us to safely rewrite genetic code without causing the cellular damage that comes with traditional cutting tools. But there was a problem. The brain is heavily guarded, and our molecular pencil is too large to fit into the microscopic delivery trucks, called AAEs, used to transport medicine into brain cells. Scientists divided the editor's blueprint into 2 halves, shipped independently in separate delivery trucks. With molecular glue added to each end, these halves act like magnetic Lego bricks inside the cell, automatically snapping together to form the working pencil. By splitting the tool into smaller pieces, researchers bypassed the brain's physical barriers and successfully smuggled a repair factory through the front door. In the mice, this worked. The 2 halves found each other and successfully corrected the A back to a G across multiple critical brain regions. This single-letter fix fully restored the missing manager protein in the brain. The behavioral change was immediate. The mice regained their physical coordination and social communication and essentially returned to normal. By fixing one miswritten letter, researchers reversed a severe neurodevelopmental disorder in a living creature. Mouse brains are tiny, so the next step was proving this split delivery method could work in the much larger and more complex brain of a primate. The results from primate trials were successful. The 2 halves navigated the brain, found each other, and assembled the editor as planned. The editor was also incredibly safe, making almost 0 accidental typos in other parts of the genome. This means we are no longer limited to just treating the symptoms of genetic brain diseases. We are entering a time where genetic destiny is no longer fixed, and life-altering typos can finally be erased.